Hi class, today we're diving into one of the most essential biochemical laboratory topics, qualitative tests for carbohydrates. We'll connect theory to practice by looking at how different sugars behave chemically and how we can identify them in the laboratory using classical color reactions. So before we start, here are our goals for this experiment. First, you'll perform a series of biochemical tests used to detect and differentiate carbohydrates. Second, you must understand the chemical principles behind each test, why a color changes, why a precipitate forms, and what that tells us about the sugar. Lastly, you'll put everything together to identify an unknown carbohydrate based on your experimental results. Think of this like solving a biochemical puzzle using chemical logic. So, what are carbohydrates? Carbohydrates are the most abundant biological molecules on Earth, and you encounter them daily, from glucose in your blood to plant starch in food. They follow the general formula CH2O, and where meaning they're basically hydrates of carbon. So chemically, they are polyhydroxyaldehydes or polyhydroxyketones, and the simplest repeat units are monosaccharides. When these units polymerize, we get polysaccharides like starch or glycogen. Although their abundance in the human body is relatively low, carbohydrates constitute about 75% by mass of dry plant materials. So, green plants or those chlorophyll containing plants produce carbohydrates via photosynthesis. In this process, carbon dioxide from the air and water from the soil are the reactants, and the sunlight absorbed by chlorophyll is the energy source. So, plants have two main uses for carbohydrates they produce. In the form of cellulose, carbohydrates serve as structural elements, and in the form of starch, they provide energy reserves for the plants. Dietary intake of plant materials is the major carbohydrate source for humans and animals. The average human diet should ideally be about 2/3 carbohydrates by mass. Carbohydrates have the following functions in humans. First, carbohydrate oxidation provides energy. Second, carbohydrate storage in the form of glycogen provides a short-term energy reserve. Next is that carbohydrates supply carbon atoms for the synthesis of other biochemical substances such as your proteins, lipids, and nucleic acids. Carbohydrates form part of the structural framework of DNA and RNA molecules. Carbohydrates are linked to lipids, which are structural components of the cell membranes. And lastly, carbohydrates linked to proteins function in a variety of cell-to-cell and cell-to-molecule recognition processes. So let's quickly recall their major functions as what I have mentioned earlier. First is as an energy source. Glucose is our primary cellular fuel. Then next is the building block. Carbohydrates serve as precursors to essential biomolecules. Next is as an energy storage. So starch in plants, glycogen and in animals. Structural role, they form part of the nucleic acids, and then cell membrane components, they consist glycolipids and your glycoproteins. So for the classification, carbohydrates are grouped based on their monomeric units and functional groups. And we can divide them into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Today, we'll emphasize monosaccharides to polysaccharides because these will appear in your qualitative tests. So, as what we have mentioned earlier, carbohydrates are biomolecules which is composed of your carbon, hydrogen, and oxygen atoms, whose functional group can either be an aldehyde or a ketone. Let's proceed first with your monosaccharides. Mono meaning one, saccharide meaning sweet. So monosaccharides are simple sugars, the most common of which is glucose. In monosaccharides, the number of carbons usually ranges from 3 to 7. Most monosaccharide names end with the suffix "ose". So if the sugar has an aldehyde group, the functional group with the structure RCHO, it is known as an aldose. And if it has a ketone group, the functional group with the structure RC, double bond O, R prime, it is known as a ketose. So depending on the number of carbons in the sugar, they also may be known as trioses, which consists of 3 carbons. Pentoses having 5 carbons, and or hexoses having 6 carbons. Monosaccharides again are the simplest sugars, therefore you can't hydrolyze them any further. So examples would include your ribose, fructose, galactose, and glucose. They differ in carbon number, functional group, as what I have mentioned earlier, either aldose or ketose, and ring structure, either furanose or pyranose. So these differences influence how they respond in each qualitative test. Let's proceed to glucose. Glucose is also called as dextrose or blood sugar. So it is the body's preferred energy source. So it's especially important for tissues lacking mitochondria, like the red blood cells. So it can be derived from dietary starch and disaccharides. So mitochondria are very important energy converters. Large amounts generate energy in cells that have limited oxygen supply, such as our eyeball. So why? The cornea is one of the few parts of our body that doesn't have blood vessels supplying oxygen. So without blood vessels, the cornea must get its oxygen directly from the air. The oxygen first dissolves in the tears and then diffuses throughout the cornea to keep it healthy. Some oxygen can diffuse through the aqueous humor within the eye, but this is slow and limited. So galactose is known as the brain sugar. It supports the brain development of infants. The monosaccharide sugar helps trigger long-term memory formation. Galactose is a monosaccharide, so when combined with glucose, which is also another monosaccharide, through a condensation reaction, the result is a disaccharide called lactose. Galactose is also a constituent of many glycoproteins, glycolipids, and your mucopolysaccharides. So galactose is also found in complex carbohydrates and in carbohydrate-containing lipids called glycolipids, which occur in the brain and other nervous tissues of most animals. A phospholipid is a type of lipid molecule that is the main component of the cell membrane. Proteoglycans are a subclass of glycoproteins with distinctive features of carbohydrate structure. Proteoglycans are primarily a type of polysaccharide. So structurally, proteoglycans are macromolecules comprised of a protein core covalently attached to glycosaminoglycans. Glycoproteins are molecules that comprise of protein and carbohydrate chains that are involved in many physiological functions, including immunity. So, is galactose converted to glucose 1 phosphate? This is the metabolism for sugars. So, galactose is one of the monosaccharides of importance in human nutrition. It is converted to glucose 1 phosphate in the liver and subsequently stored as a glycogen, or is converted to glucose and released into the circulation. So the increase in plasma glucose is known to be modest following galactose ingestion. Galactose is important structurally, as what I have mentioned earlier. It forms lactose, and lactose is used in synthesizing glycolipids, phospholipids, and glycoproteins. So inside our body, it's easily synthesized from glucose 1 phosphate. Fructose. It is a ketohexose commonly found in honey and fruits. So its ketone functional group is what makes it respond strongly in Seliwanov's test. So other things that you need to know is already presented on the screen. Next, we have disaccharides. So disaccharides consist of two monosaccharides connected via a glycosidic linkage. So we have the following examples: sucrose, lactose, and maltose. The type of glycosidic bond, alpha or beta, determines whether the sugar is a reducing or a non-reducing sugar. So disaccharides are joined by a carbon-oxygen-carbon linkage known as your glycosidic linkage. So this linkage is formed from the reaction of your anomeric carbon of one cyclic monosaccharide with your hydroxyl group of a second monosaccharide as shown in the picture. Next, we have maltose or malt sugar. It has an alpha 1,4 linkage between two glucose units. So it's produced during starch hydrolysis, and importantly, it's a reducing sugar. Maltose occurs to a limited extent in sprouting grain. It is formed most often by the partial hydrolysis of starch and glycogen. So in the manufacture of beer, maltose is liberated by the action of malt or the germinating barley on starch. For this reason, it is often referred to as malt sugar. Maltose is about 30% as sweet as sucrose. The human body is unable to metabolize maltose or any other disaccharide directly from the diet because the molecules are too large to pass through the cell membranes of the intestinal wall. So maltose is a reducing sugar. Again, please don't forget that. So if ever there is an hydrolysis, maltose will be converted into your 2-delta glucose with the presence of the enzyme or maltase. So next we have your lactose. Lactose is milk sugar. It has a beta 1,4 linkage between galactose and glucose. Because its glucose unit retains a free anomeric carbon, lactose is also a reducing sugar. Lactose is known as milk sugar because it occurs in the milk of humans, cows, and other mammals. In fact, the natural synthesis of lactose occurs only in mammary tissue. Whereas most other carbohydrates are plant products, human milk contains about 7.5% lactose and cow's milk contains about 4.5%. This sugar is one of the lowest ranking terms of sweetness, being about 1/6 as sweet as sucrose. Lactose is produced commercially from whey, a byproduct in the manufacture of cheese. So it is important as an infant food and in the production of penicillin. Lactose is a reducing sugar. again composed of one molecule of your delta galactose and one molecule of your delta glucose. So just a quick recap again, it's joined by a beta 1,4 glycosidic bond. Again, glycosidic bond is the bond from the anomeric carbon of the first monosaccharide unit being directed upward. So the two monosaccharides are obtained from lactose by acid hydrolysis or the catalytic action of the enzyme lactase. So many adults and some children suffer from a deficiency of lactase. So these individuals are said to be lactose intolerant because they cannot digest the lactose found in milk. A more serious problem is the genetic disease galactosemia, which results from the absence of an enzyme needed to convert galactose to glucose. Certain bacteria can metabolize lactose, forming lactic acid as one of the products. This reaction is responsible for the souring of milk. Next is sucrose. Sucrose is table sugar. It's unique because both anomeric carbons are involved in the glycosidic bond. That's why sucrose is a non-reducing sugar, a very important concept in the tests later. Sucrose, probably it's the largest selling pure organic compound in the world, is known as beet sugar, cane sugar, table sugar, or simply sugar. Most of the sucrose sold commercially is obtained from sugarcane and sugar beets, whose juices are 14% to 20% sucrose. So by evaporation of the water and recrystallization, the dark brown liquid that remains after the recrystallization of sugar is sold as molasses. So the sucrose molecule is unique among the common disaccharides in having an alpha 1 beta 2 glycosidic or head-to-head linkage, because this glycosidic linkage is formed by the hydroxyl group on the anomeric carbon of a delta glucose and the hydroxyl group on the anomeric carbon of beta D fructose. It ties up the anomeric carbons of both glucose and fructose. And sucrose is a non-reducing sugar. Next, we have polysaccharides. Polysaccharides are large carbohydrate polymers. Examples are starch and glycogen. Their structure, linear or branched, determines how they react with iodine test later. The polysaccharides are the most abundant carbohydrates in nature and serve a variety of functions, such as energy storage or as components of plant cell walls. Polysaccharides are very large polymers composed of tens to thousands of monosaccharides joined together by glycosidic linkages. So the three most abundant polysaccharides are starch, glycogen, and cellulose. These three are referred to as homopolymers because each yields only one type of monosaccharide after complete hydrolysis. We also have the term heteropolymers, which may contain sugar, acids, amino sugars, or non-carbohydrate substances, in addition to monosaccharides. Heteropolymers are common in nature, such as your gums, pectins, and other substances, but will not be discussed further on this lesson. So the polysaccharides are non-reducing carbohydrates. They are not sweet tasting and do not undergo mutarotation. So starch, the major storage polysaccharide in plants, has two components. So we have the amylose, which is a linear helix, and the amylopectin, which is branched in structure. The helical structure of amylose is responsible for the blue-black iodine reaction. Starch is the most important source of carbohydrates in the human diet and accounts for more than 50% of our carbohydrate intake. So again, it occurs in plants in the form of granules, and these are particularly abundant in seeds, especially the cereal grains and tubers. So where they serve as a storage form of carbohydrates. So the breakdown of starch to glucose nourishes the plant during periods of reduced photosynthetic activity. So we often think of potatoes as a starchy food, yet other plants contain a much greater percentage of starch. Potatoes only have 15% of starch, wheat 55%, corn 65%, and rice 75% of starch. Commercial starch is a white powder. So again, just a quick summary. Starch is a mixture of two polymers, amylose and amylopectin, and then the complete hydrolysis of your starch yields in successive stages glucose, then starch, then dextrine, then maltose, and lastly glucose. So in the human body, several enzymes known collectively as amylases degrade starch sequentially into usable glucose units. So this is just the comparison between the structure of your amylose and your amylopectin. Next, we have your glycogen. Glycogen is a storage polysaccharide in animals. So it's highly branched with alpha 1 to 6 branch points in every 8 to 12 glucose units. Because of this structure, iodine produces a reddish-brown color instead of blue. So glycogen is the energy reserve carbohydrate of animals. Practically all mammalian cells contain some stored carbohydrates in the form of glycogen, but it is especially abundant in the liver, 4 to 8% by weight of the tissue, and in skeletal muscle 0.5 to 1%, as what I have mentioned earlier in this video. So like starch in plants, glycogen is found as granules in the liver and muscle cells. So when fasting, animals draw on these glycogen reserves during the first day without food to obtain the glucose needed to maintain metabolic balance. So again, glycogen is structurally quite similar to amylopectin, although glycogen is more highly branched, 8 to 12 glucose units between branches, and the branches are shorter. So when treated with iodine, glycogen gives a reddish-brown color. Glycogen can be broken down into its delta glucose subunits by acid hydrolysis or by the same enzymes that catalyze the breakdown of starch. In animals, the enzyme phosphorylase catalyzes the breakdown of glycogen to phosphate esters of glucose. About 70% of the total glycogen in the body is stored in muscle cells. Although the percentage of glycogen by weight is higher in the liver, the much greater mass of skeletal muscle stores a greater total amount of glycogen. Next, we have aldolases. Aldoses rather. Aldoses contain an aldehyde group. So examples include ribose, glucose, and galactose. So this aldehyde group is responsible for their strong reducing ability. Next, we have ketoses. They contain a ketone group like fructose. Their activity differs from aldoses, especially in tests like Siliwanov's. So monosaccharides cyclize to form rings. So we have pyranose. It is a 6-membered ring. Example, we have your glucose, while a furanose is a 5-membered ring. Example, we have your fructose. So these ring forms affect the dehydration reactions in Bial's and Siliwanov's tests. Reducing sugars have a free anomeric carbon that can reduce metal ions. Examples include all monosaccharides such as your maltose and lactose. And then you'll see them turn positive in tests like Morse or bar foods. A reducing sugar is a carbohydrate that is oxidized by a weak oxidizing agent. When we talk about a weak oxidizing agent, it's an oxidizing agent capable of oxidizing aldehydes but not alcohols, such as the Tollens reagent. So in basic aqueous solution, the characteristic property of reducing sugars is that in aqueous medium, they generate one or more compounds containing an aldehyde group. So the reducing sugar, such as glucose and fructose, have a free aldehyde group and ketone in their structures respectively. So the reducing sugar mostly forms a hemiacetal structure where a carbon gets attached to a couple of oxygen atoms, thus resulting in the formation of either ether or alcohol. The reducing sugars can be oxidized with some relatively mild oxidizing agents such as salts of metals. In an aqueous solution, so the reducing sugars or the reducing agents generally generate one or more compounds comprising an aldehyde group. So in the Maillard reaction, so the reducing sugars react with the amino acids and a series of chemical and biological reactions occur. The Maillard reaction is the process in which amines react with the reducing sugars, resulting in the browning of the food. So the reaction normally occurs when either the food is left at room temperature for a long period or is heated. So the browning of food can be often seen on the crust of the bread or the skin of roasted dietary items. So similarly, the reducing sugars produce mutarotation and form ozones. So non-reducing sugars, so these sugars do not have a free anomeric carbon. So examples, we have sucrose, starch, and glycogen. So they will test negative in many redox based carbohydrate tests. The sucrose molecule is unique among the common disaccharides in having alpha-1 beta-2 glycosidic head-to-head linkage, because this glycosidic linkage is formed by the hydroxyl group on the anomeric carbon of a delta glucose and the hydroxyl group on the anomeric carbon of beta delta fructose. So it ties up the anomeric carbons of both glucose and fructose. Now let's proceed to the Morse test. This test detects reducing sugars. So under strong alkaline conditions, reducing sugars undergo aldol condensation, leading to caramelization. So a positive result gives a caramel odor and yellow to brown coloration. Reducing sugars like glucose, fructose, ribose, and maltose turn positive. So these images show how reducing sugars produce a brown to yellow or brown-yellow complex. Non-reducing sugars remain unchanged. So we have the Barfoed's test. You can see the characteristic red precipitate for positive monosaccharides here. It's found on the left side of this photo. So Barfoed's test is a test for the presence of monosaccharide. So the principle, if strong reducing sugars like monosaccharides are oxidized by your copper ion in acetic acid solution to carboxylic acid, it will form the cupric oxide precipitate within 3 minutes. And the positive result is, of course, the presence of brick red precipitate. So this summarizes the results. For glucose, fructose, galactose, and ribose, they will turn positive, while other sugars, they are negative. Now let's proceed to your Bial's orsinol test. So this test detects pentoses, which dehydrate to form furfural. So furfural reacts with your orsinol and ferric ammonium ions to produce a bluish green color. So hexoses dehydrate more slowly and produce yellow-brown colors instead. So pentoses, they will produce a bluish-green product, while hexoses will give you a yellow-brown product if that's the positive reaction. So observe how pentoses differ significantly in color from hexoses. Next, we have the Seliwanov's test. This test differentiates ketoses from your aldoses. So ketoses dehydrate rapidly in hydrochloric acid plus resorcinol, producing a bright cherry red color. So aldohexoses react slowly. They will produce pale pink coloration, while sucrose also gives a red reaction because it hydrolyzes to glucose plus your fructose. So these sample results compare fructose, which has a strong ketose reaction, and then glucose is slow reaction having pink coloration or pale pink, and glycogen will have a negative result. Next, we have your iodine test. So this test detects polysaccharides, especially those with coiled structures. So we have different products actually. So for starch or amylose, they will have a blue-black coloration. For glycogen, reddish-brown. Dextrins, they will have reddish-purple. And iodine stays yellow-brown when no polysaccharide is present. So this is what I have mentioned earlier. So dextrin or dextrins are a group of low molecular weight polymeric carbohydrates produced by the hydrolysis of starch or glycogen. So please do remember this, everyone. So this test is highly specific for polysaccharide in solution. So your cellulose does not give this test, but you will not find cellulose in the solution. And then the color disappears on heating and reappears when the solution is cooled down. So if the color does not appear upon cooling, it indicates that iodine has vaporized during heating. So please be careful. Don't add too much iodine to prevent false results. Okay. So next, these images show the expected colors for starch, glycogen, and dextrins. For starch and glycogen, the starch will yield a blue-black coloration, while the glycogen will yield a reddish-brown coloration. For ribose, glucose, fructose, and lactose, they will have a negative result. Next, we have the Mukic test or the Music test. So this is a specific test for galactose. So galactose, when oxidized with your nitric acid, it will form an insoluble mucic acid crystals or mucic acid crystals. Because very few sugars from these crystals, this test is a great confirmatory tool for the presence of galactose. So this presentation or this slide shows crystal formation of a typical of a positive mucic or the mucic acid reaction. Of course, galactose and lactose will have a positive result, while other sugars will have a negative result. And with that, everyone, thank you so much for listening. I would like to conclude my discussion about experiment number 9 for qualitative test for carbohydrates. I hope that you have learned something from this video. Thank you so much and have a good day.